Hey, 7th graders, this is the pond water lab, and this is going to be a 2-day lab. So if you guys are gone both days, obviously you guys are going to watch this video. You're going to do this whole assignment. You guys will be able to collect data from home using the other videos that I've linked throughout. If you guys are just gone one day, I just kind of want to break it down for what the days are going to look like. So for the most part, the first day of the lab, if you guys are gone through that, we're going to kind of cover all of this information that you're listening to in this video, or most of the information in this video, and basically materials from slides 1 through about 22 where we do the control data, okay? If you guys are gone day 2 of this, that's where you guys are going to be doing most of the actual lab work, and you guys will be collecting the data. So you guys will be finishing up slides 23 and then through the end of the assignment where you guys collect the data. Again, you'll be able to do that from home just by watching the videos, copying in your data table stuff, and then answering the questions at the end and making predictions. So this is again the pond water lab. Your learning targets, I can define and give an example of an abiotic factor. So in class a lot we've been talking about food chains and how different organisms can affect the size of other populations based on that food chain. So one example of our food chain that we've been referencing a lot and will keep referencing a lot is that whole like sea urchin, green kelp, the moon jellies, the orca, the sea turtles, all of those. So what we've seen so far is, you know, if we change the number of moon jellies, then we're going to see effects with the other living organisms in that same food chain. So we might increase the number of orca, or we might decrease the number of sea turtles, or whatever the case may be, okay? That's really focusing on how those biotic or living factors change other living factors. And again, we've talked about that a lot between talking about our community interactions, predator-prey, competition, and symbiosis, where you guys saw, you know, mutualism, where both species have a relationship that benefits both of them. Commensalism, where one species benefits, the other species isn't harmed at all, so like a honeybee or a barnacle. Sorry, honeybee's a bad example. That's actually mutualism. A barnacle and a whale, the barnacle gets to move around and gets fed, but it doesn't hurt or help out the whale to be attached to the whale's body. And then parasitism, where it isn't quite predator-prey because that parasite doesn't want to kill its host organism. It needs its host organism to be alive in order for it to benefit, but the host is, of course, harmed in this parasitic relationship. So those are all relationships where we look at relationships between living things, okay? On the other hand, though, that's not the only thing that's going to affect a number in a population. So we're going to really start talking about this concept of abiotic factors today. Okay, so an abiotic factor, just like biology is the study of life, a biotic factor are all the living things in an ecosystem. An abiotic factor are going to be all the non-living things in that ecosystem that actually make a difference to the organisms that live there. And we'll talk a little bit more about that in a second. And then we're going to focus in on some specific abiotic factors for aquatic ecosystems. So your other learning target, I can explain how an abiotic factor such as temperature, pH, salinity, and nitrate concentration would affect the size of populations of different organisms in 3 different aquatic environments. Okay, so moving on slide 2, you guys have your warm-up. So I'm going to have you guys go ahead and pause this video, fill out your warm-up, and then when you guys are done with slide 2 and your warm-up, I want you guys to come back to this video. All right, so you guys should have just completed your warm-up. Questions 1 and 2 are really good review questions for us. We've been asking this question and talking about it this whole unit so far, right from the very first lesson in 1.2. Okay, so what are 2 ways a population can increase in size? The main thing what we talked about is there's more births than deaths, and there's a million different reasons that you guys might see more reproduction and more births than you guys see deaths. For instance, there's plentiful food. Okay. The other reason you might see a population increase in size, remember that when we talk about a population, we're talking about all the organisms of the same species that live in a specific area and can kind of intermingle and maybe even reproduce in that area. So if we talk about migration, animals might be immigrating into that area, and that would increase the population size. Okay, on the other hand, what are 2 ways a population can decrease? It's just the opposite of that. Animals might move out, and again, they might move out for various reasons. Maybe there's just not enough food, so they have to leave and go somewhere else to find food. Okay, or it could be something like more deaths than births, and in that case, again, there's many causes for more deaths than births. Maybe hunters move in. Okay, maybe the predators change in that area. Maybe there's a big natural disaster that wipes out all their food source. Okay, and then we might see more deaths and births. Okay, so again, think about what those, when you guys list those answers, what does it really mean? Okay, more deaths and births, more births and deaths, and migration. Okay, last but not least, in your guys' OneNote, you guys should have had the answer to this question: what is an ecosystem? So if you guys looked in your OneNote, you guys will see that it's all the living and non-living things that are kind of together in an area. And so that's where we're really going to focus on that abiotic factor today, looking at all the non-living things. So that leads us into our next question, or our next little vocab answer, if you will. So an abiotic factor is all the non-living parts of an ecosystem that influence how successful a population may be in that ecosystem. Okay, so one thing that we talk about is climate is huge. Okay, weather. We're not going to see polar bears living in Hawaii because they're not adapted to that climate. Okay, we're not going to see cactuses living in the Arctic because they're not adapted to that environment. So what I want you guys to do here in a second is I want you guys to just grab a piece of scratch paper, and I want you guys to take about 2 to 3 minutes at home, and I want you guys to jot down as many different abiotic factors, these non-living things that we would find in an ecosystem that would influence kind of the populations and how big of a population might be able to live in a specific ecosystem. So go ahead, pause this video, take about 2-3 minutes and brainstorm as many different abiotic factors that you guys can think about, and then come on back to this video. All right, so we sat and we brainstormed in class as well, and we came up with a big old list of abiotic factors. If we're just talking about a little bitty rock on the ground, okay, that's definitely a non-living thing, but does it really influence what organisms live there, okay? A bear isn't going to come up to that teeny tiny rock and be like, woo, I can't live here, this isn't suited to me. But if we're talking about like a steep terrain of a mountain, that's definitely going to influence the kind of organisms that can survive in that terrain. So terrain would be a really good abiotic factor. Okay, other things we talked about is water supply. Is it readily available or no? And how do we adapt to that? Shelter. Okay, what different options do we have for shelter out of the elements, away from predators, whatever the case. Terrain is huge, like we just talked about. Only certain animals can live on some crazy terrain, and vice versa. Okay, sunlight, that can apply to plants, but it also can apply to animals if you're looking at cold-blooded species that depend on heat and sun for them to maintain their body temperature. Temperature and precipitation both kind of look at climate, so how much precipitation does it get, what kind of precipitation if it gets. So if we're looking at a desert could be hot or it can be cold. If we're looking at Antarctica, Antarctica is actually considered a desert because even though it gets a lot of snow, that snow isn't usable precipitation. Okay, so you know, that still is considered a desert because we don't have usable precipitation in the form of that snow. Okay, we could look at soil. Is it sandy soil? Is it rich soil? Are we able to dig in this soil? Okay, can we find shelter in that soil? Are we going to grow a lot of plants, which mean food in that soil? Those kinds of things. You know, if it rains, does that soil absorb the water, or does it just run right off? Okay, we could look at elevation. The higher we go up, the less oxygen it is. So that's important to what organisms can survive there. If you guys were paying attention for the elk and wolves and Yellowstone case study activity that we did, thermal activity is really big. Okay, it definitely affects the organisms that live there. If we're looking at aquatic, maybe the amount of salt. Okay, and then a big one for all ecosystems is human impact. So are we looking at a lot of litter, a lot of pollution, those kinds of things? That definitely affects population. Okay, not a naturally occurring one, but one no less. Okay, today we're going to be talking about 3 different aquatic ecosystems, and we're going to be looking at 4 different abiotic factors to test in those 3 aquatic ecosystems. So we're going to look at temperature, pH, salinity, and nitrate concentration. So obviously you guys know what temperature is. But what we're going to see with temperature of an aquatic ecosystem like a pond or a lake or an ocean is that's going to vary seasonally. If it's winter, obviously you're going to have a lower temperature. If it's summer, you're going to have a little bit higher temperature in your water. And also the deeper you go in that body of water, you'll see differences in temperature. So what we see overall is organisms can survive in a wide range of temperatures. Not all organisms. If we look at little trout minnows, if you guys take those out and put them in just regular room temperature water or a fish tank, they're going to die because it's just not cold enough for them. Okay, what we see is the colder the water, the more oxygen in that water. Okay, and obviously they need that water to survive. So While a lot of aquatic ecosystems can survive in a wide range, a lot of them can't as well. And then we might even see some organisms that need a specific temperature in order to reproduce, depending on how they reproduce. Maybe they need a certain temperature to be good at making eggs, whatever the case is. Okay, so that's definitely a factor that affects what populations can live in an aquatic ecosystem. pH, this tells us how acidic or basic water is. So if we're looking at our scale, it goes from 0 being the lowest, 14 being the highest. If we're going from 0 to 6, that's what we call a low pH, and we consider those acids or an acidic environment. Okay, above 7, 7 is neutral, so if we're looking at water, water should be neutral, it's right at 7. And if we're looking above that in our 8 to 14 range, that's going to be basic or alkaline. You guys might hear both of those used, and that's going to indicate a high pH. Okay, few species can live in acidic or basic aquatic environments. So really they need that neutral range to be able to survive. Okay, somewhere in between 6 and 8 generally. But again, it just depends. Even 6 or 8 might be too acidic or basic. We need basically water that's neutral. Here's a little example of pH. So you got batteries is one of our lowest acids that we can find. So it's really acidic. On the other side, like something like drain cleaner, which dissolves all the crap and junk in your guys' pipes but doesn't dissolve the metal itself. That's super basic. A lot of your cleaning solutions, soap, ammonias, that smell you guys get in Windex, Tums and Rolaids or stomach tablets, they're basic and their purpose is to neutralize an acid. Baking soda, blood's even a little bit basic. Water's right here in the neutral area, 7. When we think pH, we don't actually have a unit label for that. We just use the number. You guys can see milk, coffee, stomach acids, about a 1 or a 2. Lemon, the citric acid in lemon, it's what gives it that sour taste that is acidic. So a lot of that sour tasting stuff is usually an acid. That's why like vinegar is sour tasting. Lemons are sour tasting. All citrus fruit has citric acid, so they're sour tasting. Even stomach acid has got a sour taste to it. Okay, so that's one thing. Salinity is how salty the water is, and what we see with organisms is they're either adapted to live in fresh water or they're adapted to live in salt water. There's very few organisms that can go back and forth between fresh or salt water. Okay. We are going to test salinity today using a hydrometer. What we know, if you guys have ever swam in the ocean before, the ocean's obviously a saltwater environment, is you guys are more buoyant in salt water, meaning you float easier. Okay, so if you guys have ever swam in the ocean, it's a little bit easier to swim there because again, your body floats a little bit better. Okay, so we're gonna use this little hydrometer and you'll fill it up with water, and what you'll see is this little arm, this little white lever in there is going to float if there's a lot of salt water. So you guys will be able to see how much it floats. We're going to kind of look at the numbers on the outside of this scale, and that's going to be measured in parts per thousand. So again, more salinity it has, the more this little lever is going to float, and it's going to point to the higher number in parts per thousand. Okay, so that's going to be how we measure salinity. And then finally, nitrate concentration. So there's going to be a little bit that we learn about nitrate. Nitrates are a chemical found in fertilizer, and obviously we use fertilizer to make plants grow. That being said, nitrogens are rich in nitrogen, and that's what the plants really like when they are looking at higher growth. So they're rich in nitrogen, which helps plant life grow, such as algae. But if you think about it, you're like, yay, more plants. That means more oxygen, right? Because plants give off oxygen. But that's not always the case, especially in an aquatic ecosystem. So if we're looking at algae, to some extent, algae is important in an aquatic ecosystem. But just as anything, too much is not always a good thing. And so if we have too much algae, which we would call an algal bloom, we're going to see that affect the amount of oxygen in an aquatic environment negatively. So algal blooms affect oxygen levels in the water. Only some organisms are adapted to be efficient enough to live in environments with low dissolved oxygen. So I want you guys to take a second on slide 11. I want you guys to pause this video and watch this video about how algae and having an excessive amount of nitrogen can actually lower the oxygen in an aquatic environment. So go ahead, pause, watch this video, and then come on back when you're done. All right, so you guys should have a little bit more of an idea of why having nitrogen and fertilizer in our water is bad and how that increases the algae and then lowers our oxygen, okay? What we're going to be doing today is you guys have pond samples from 3 different aquatic environments, and I'm just going to talk to you guys about those environments right now. So Aquatic Ecosystem A is going to be Lake Erie. So what we know about Lake Erie is one of the great, is one of the Great Lakes located between Canada and the United States. So it's around Pennsylvania, Ohio, that little area up in there. Fertilizer from nearby farms has run off into the lake, increasing the level of nitrogen and phosphorus, which we just learned about what that does to that ecosystem. Increased levels of nitrogen and phosphorus encourages the algae to bloom in this aquatic environment. So here's a little map of Lake Erie. You guys can see here's Canada, Ontario in there, Michigan, Ohio, Indiana, Pennsylvania. So you guys can see where Lake Erie is. And this is actually a picture of the algae blooms. So all this green stuff is the algal blooms in Lake Erie due to that presence of nitrogen. Okay, our second ecosystem is just any old shallow pond in Massachusetts. So what we know is there's several large cities around it. And then acid rain is really common in this environment. So again, another little short video for you guys to watch. So I'll have you take a second, pause this video and watch the what is acid rain video and then come on back. Okay, this is just a generic photo of what a pond might look like. Okay, this shallow pond in Massachusetts. And then finally, Aquatic Ecosystem C is the Aral Sea. It's located between Kazakhstan and Uzbekistan, and it used to be the 4th largest lake in the world, but then shrunk because there was 2 rivers that fed it, and they diverted those rivers to use for farming. And so they no longer had that feed of water coming into this lake, and it shrunk a lot. And because it shrunk, it wasn't getting kind of a freshwater supply to it, and it became very saline, so a lot of salt in it now and minerals. Okay, so this is Kazakhstan over here. You guys can see Russia, China, Kyrgyzstan. Here's Uzbekistan. So this is our Aral Sea that kind of goes over the border of both Kazakhstan and Uzbekistan. And this is what the Aral Sea used to look like, and now this is what the Aral Sea looks like. So you guys can see how much that shrunk. It's a huge difference. Okay, this whole area out here used to be the Aral Sea. Okay. So what you guys will be doing, that kind of encompasses most of day 1, but what you guys will be doing is, obviously in class we'll be wearing safety goggles, and you guys will be measuring water samples. I obviously made them in a lab, so they're not actual water samples from these places because I don't have time for that or the money for that. So you guys will be collecting data on the temperature, the pH, the nitrate concentration, and the salinity for these 3 ecosystems. Okay, here's how you guys measure is obviously temperature. You guys are going to see me measure the temperature using the little probe and recording it in degrees Celsius. pH, I'm going to use litmus paper. I'm going to dip the strip in, and then I'm going to look at the little key on the package to see what the pH is. Remember, temperature, we're going to do degrees Celsius, so that would be your label. pH doesn't have a label. You guys just include the number between 0 and 14. Nitrate concentration, you guys are going to use milligrams per liter. Again, you have just a little test strip that you guys are going to see me use, and then compare it to a color key very similar to what we did in pH, but again, milligrams per liter, we would write it like this, little m, little g, and then a slash, and then an uppercase L. So that's going to be for your nitrate. This is degrees Celsius, and this one is going to be salinity. So I'll show you guys again how to use this, the little hydrometer to do salinity. Okay, and we're going to measure that in ppt or parts per 1,000, so all lowercase there. So let's go ahead and take a look at the control data. You guys have a little video to watch. It's going to give you guys the control data, and what I do is I go through and I test just plain tap water. Okay, and there shouldn't be a whole lot in our tap water. There's a little bit, not a whole lot in our tap water. If I use like bottled water that was purified, then I shouldn't have anything in there. So go ahead, take a look at our control data. So watch this video here, right click it, open it up, pause this video, collect your data for your control, and then once you're done, we'll move on to Ecosystem A. All right, so for Ecosystem A, you're going to do the same thing. You guys have a little video for you guys to watch, but how this is different than the control is obviously we're doing temperature, pH, nitrate concentration, and salinity. We also have data for 40 years ago. So you guys are going to watch this video, you're going to collect data, and you're going to record it right here under current data. If you guys see a change between the temperature then and the temperature 40 years ago, or excuse me, the temperature now and the temperature 40 years ago, that you guys think is significant, what I want you guys to do is take your draw tool and circle that. So if I measure the temperature for ecosystem A and it comes up as like 42 degrees Celsius, okay, then you guys would put a circle around temperature just to kind of take a note and indicate, all right, something's different with the temperature from 40 years ago to now. So that's something I need to pay attention to when I'm answering my questions at the end. Okay, so look through after you guys collect data for ecosystem A, look through and circle any that you guys think are significantly different. Okay, there's a pretty big difference in maybe the temperature, pH, nitrate concentration, or salinity. Okay, do the same thing for B. You guys will pause this video, watch the data collection video for ecosystem B, record your data, compare it to 40 years ago, and again, take your draw tool and circle any of these that might be significantly different from now compared to 40 years ago. And same thing with ecosystem C or Aral Sea. Watch that video, record your data, and then compare now to 37 years ago and circle any large differences you had. Once you guys have done that, you're going to go ahead and take a look at 3 organisms and kind of make a prediction as to what you guys think is going to happen to those organisms in these ecosystems. So examine the data about Cyanobacteria. So Cyanobacteria obviously is a little type of bacteria that looks, sometimes people call it blue-green algae. Based on the changes to the water in ecosystem A, so for this one you guys are just focusing in on ecosystem A, that you discovered in part 1, explain whether you think the cyanobacteria population in ecosystem A will increase, decrease, or stay the same. Describe the evidence that makes you think this. So you're going to take a look at this data here that says cyanobacteria survive best in this range. So their temperature is 20 to 30 degrees. Their pH is about a 7 to an 8.5. Nitrate concentration is no known range, but the nitrogen in nitrogen is an important nutrient for cyanobacteria, so kind of like the algae, it would grow a lot. And then salinity 0 to 20 parts per thousand. So make predictions on that in the form of this. So predict what will happen to the cyanobacteria in ecosystem A. Do you think it's going to increase over that 40-year time period, decrease, or stay the same? And then describe what evidence. So when you describe the evidence, I should see data and numbers from your lab, and then write about 2 to 3 sentences explaining that data and that evidence that you're coming up with. So again, I should see numbers in your lab or in your explanation. Okay, next organism you're looking at is the yellow perch. You guys are focusing specifically on ecosystem B, so that shallow pond. Explain what you think the yellow population or the yellow perch population will do, whether it's going to increase, decrease, or stay the same. We know that yellow perch survive best in this temperature range. In a pH that's a little bit, it can be a little bit more acidic, so 4 to 9.5. We don't really know much about the nitrate concentration, and then salinity is going to be pretty low, so they really can't survive in a salty environment. Okay, so low salinity on that, so make your predictions about what's going to happen to the yellow perch, increase, decrease, or stay the same, and then again describe that evidence that helps you support your answer here, whether you guys think it's going to increase, decrease, or stay the same. As per usual, I should see numbers from your lab, okay, numbers from your data tables, and you should use about 2 to 3 sentences to explain that. Finally, your last organism is this xander, which is a fish, and again, you guys are focusing on ecosystem C, okay? We know that it survives best in a temperature from 10.4 to 26.9. pH range is going to be 6.5 to 8.2. Nitrate concentration is going to be unknown. We don't know much about that. And then salinity needs to be pretty low for this fish to survive. So again, make your predictions about this, what's going to happen, and then give me specific numbers and evidence from your data tables above to explain that. And then once you guys are done with that, you're going to go ahead and hand it in. If you have any questions, let me know in your guys' wrap-up, and I hope you guys are having a great day. Again, this is a 2-day lab. I'll talk to you later. Bye.